Good
Sir,
morning, all of you.
I
I
would
hope
like to introduce Raghuveer Singh Parmar. Sir has pursued M.Pharm in Quality Assurance from Gujarat Technological University and currently is associated with Sun Pharmaceutical Industries Limited. He has more than 9 years of working experience in pharma industry, and during this tenure, he is actively exposed in pharma manufacturing, QA, QC, and analytical development related activities. He is involved in CGMP, analytical and regulatory training for academic students and the professionals in pharmaceutical industry by delivering various seminars. So with this brief introduction, I welcome all the audience, teachers, on behalf of Paralist of Pharmacy in today's webinar, Advanced Instrumental Techniques for Drug Analysis. I invite Mr. Raghuveer Parmar to deliver a talk. Over to you, sir.
okay. Thank you very much, madam. I can see that it is Facebook Live, so the students might have joined the Facebook Live as I hope audio will be so I would like to start with advanced instrumentation techniques for drug analysis. Uh, Friends, this is one of the most important aspects of the pharma industry. Let me tell you that we are going to discuss today the aspects without which there is no other possibility for the pharma industry to run, whether it is a pharma manufacturing or it's a pharma research and development. It is not at all possible for the pharma industry to run without the instrumental technique and drug analysis. We'll have overview one by one what instrumentation techniques right now the industry is using. And as a B Farm students or the pharmacy students, I would definitely I also passed from this situation actually. When we study, no, we don't have the exact importance or exact role of the instruments, but when we become a stakeholder of the pharma industry, when we get a job or when we are involved in pharma companies, we get to know that how these instruments and analytical techniques is important to us. Okay, so without wasting much time, let us begin with that point of with that point in our mind. So actually when we study the instruments in theoretical way, I hope the slide have changed. Okay, yeah.
Yeah, it's changing.
Okay, because in Facebook live it is not changing, that's why. Okay, okay, yeah, it's okay. So we will try to cover today the instruments and that is latest instrumentation techniques that is used in pharma analytical laboratory. The analytical laboratory may be of quality control laboratory or the analytical development department of the research and development unit of the pharma company. So as a as an analytical perspective, if we see what is the role of analytical technique, if we understand this particular thing in a layman language, it would be like we want to know what is there inside the product, right? Suppose for example organic synthesis department, which is synthesizing the active pharmaceutical ingredients or impurities or whatever it may be, but chemically synthesis done by organic synthesis department. And after synthesizing the molecule, they want to confirm whether the reactions have been taken place or not, the product they are claiming have actually been formed or not, right? Because they 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 perform reactions based on their knowledge, based on the chemistry, but actually the same chemical reactions that they have expected have been taken place or not. How can they confirm? There is no other way than utilizing the analytical techniques. And that's why friends, analytical techniques is so important, so that without using that, no scientist, whether it may be organic synthesis scientist or maybe formulation development scientist, no scientist can claim that the product they have formed, the product they have synthesized, the product that they have formulated have been actually formed, right? This is one aspect. So this is one aspect, is knowing things inside the product, and that is the role of instrumentations. Now we'll go little bit deeper into the topic. How can we know, and what things needs to be known for the scientist, right? Or for the estimation, if we talk in terms of quality control rare, see, quality control unit just have to estimate the things. They don't have to face new things, but they have to estimate the existing things. They know that the product must contain the product, but in what amount, what is the percentage assay of the drug, right? So they are knowing that the drug is present, but actual content or the estimated content they don't know, and therefore they use the analytical instruments to identify the exact estimation of the product, right? With this, we were discussing that what things should be known, right? So, and based on that, the analytical instruments have been classified and have been used in the industry, right? Suppose we'll take example one by one, and we'll try to understand how this is happening, right? If we talk about the API, first of all, what things, the what things to be known? First of all, the content, I mean the main active ingredient of the API is correct or not. First thing is that quality, and then content, right? For example, if we talk about, say, product is bivalirudin is one API. So suppose organic synthesis have synthesized the bivalirudin molecule. So really bivalirudin have been synthesized or not? First thing is that. Next thing, if bivalirudin is correctly synthesized, in what amount bivalirudin is present in percentage? Suppose if I weigh 100 gram of that powder, how much how much gram of the bivalirudin is present, and how much percentage of the impurities or other relevant products is present in the powder, right? So that is quantity. So first is quality, second is quantity, right? So this is the things that needs to be known. Now let us come to the analytical point of view. If I want to know the quality of the product, there are various techniques. If I want to know the quantity of the product, there are various different techniques, right? Say for example, if I want to know the quality of bivalirudin, I mean really the bivalirudin have been formed, there are various ways which I can use, right? Suppose for example, nuclear magnetic resonance spectroscopy is there, which can say that based on the carbon and protons present in the structure, the instrument will tell us based on spectra reading, the scientist will confirm that yes, exactly the bivalirudin spectra is matching with the structure, chemical structure of the bivalirudin, right? So it is again interpretation part, but as of now we'll understand that the chemical structure can be confirmed. Chemical structure of the any compound can be confirmed by nuclear magnetic resonance spectroscopy. One more technique in identification is IR spectroscopy, where we can confirm the specific functional groups in the structure of the chemical compound. Say for example, ketone functional group, amide functional group, any functional group have specific band in the IR spectra, and thereby so NMR, IR, then LCMS. Knowing the molecular mass of the compound is also one of the identification tests, where we can confirm that yes, the molecular mass of bivalirudin is, say for example, I'm not saying the right exact molecular mass, but say for example, take the molecular mass of bivalirudin is 2000. So the 2000 mass must be present in LCMS spectra, right? Otherwise, LCMS is also very specific technique, because it will give the exact 2000 mass to the scientist that yes, the compound that you have given have this 2000, not 1999 and not 2001, not a difference of one. Exact 2000 mass must be present, because it is bivalirudin. If I am getting mass of 2001, means it is not bivalirudin, it is something else, right? So LCMS can also be used in this way to qualitatively determine the presence of the active ingredients. Now let us come to the next point. So basically we utilize in industry NMR, IR, and LCMS. These are the major three instruments that we can use. Okay, so used to confirm the structure or to have references in which we can confirm the structure. There are instruments like CHN analyzer or XRD diffractometer. They have also some purposes, but widely used instruments are IR, NMR, and LCMS. CHN analyzer can also tell the amount of carbon, hydrogen, and nitrogen in the structure. And in suppose in XRD, what is the role of XRD? We can, if you know that some compounds have different forms in the structure, suppose crystalline form or amorphous form. So that kind of forms can be confirmed using XRD diffractometer. Okay, so we have talked this the instruments used for qualitative determination. Now let us come to the next point. What if I have to measure the exact quantity? I know that bivalirudin is present in the compound. Okay, I know it is the major compound, but how much percentage of bivalirudin is present in the compound? That I need to know, and how much amount of the impurities is present. So friends, here comes a play role. Here comes the role of chromatography, right? Now it's a huge, subject. Chromatography really helps industry to identify and quantify the structure, right? So I will run the bivalirudin chromatogram. Um, Before that, development of method would come for those students who are really basic at the B farm level. They would not know much about the exact detailed method development and other aspects of the analysis part, but let me brief you that analytical development of method means, see, these are the instrumentation techniques. These are really sophisticated instruments. So it does not mean that they are perfectly automatic, right? Automation is there, but we have to we have to specify some parameters specifically for some product. Say for example, we are sticking to bivalirudin only. Suppose bivalirudin is example of the drug. So the specific instrument parameters required for this compound is different. So every product to product different parameters would be used. For example, if we talk about chromatography, liquid chromatography, then the mobile phase preparation, pH of the mobile phase, the column that can be used, the flow rate that can be set, the column temperature, the detector wavelength, these are the parameters which are highly specific to some drug. If we take one drug, these are the one set of parameter. If we take another drug, there are different set of parameter. If we take third drug, they would be specific parameter. So analytical development scientists have to work on that, and they have to specify that if you want to estimate the exact quantity of this compound, then these are the set of parameters that you have to use for this instrument, and it is called the method, analytical method, because it contains the details of method, how we can use that instrument to estimate the content. Now I hope this is clear for analytical method. So we were talking about quantitative estimation. We have identified the quality of the material using qualitative techniques. Now we are going to quantitative estimations. How can we do that? Using chromatography. Suppose, say for example, we have developed the method, we have set the parameters, that these are the parameters which can be used for estimation of the drug using HPLC. So what chromatography will do? Chromatography would elute the compound, right? I have to prepare the solution of bivalirudin and inject into the HPLC, so that along with bivalirudin, if some impurities are present in the material or some other products, relevant products or irrelevant products are present in the compound, they would also run with the bivalirudin and with the column, right? So mobile phase is moving from injector to column, and through the column they come out into the detector. So what detector will do? Detector give, so the illusion pattern would change, right? The meaning I want to say is bivalirudin illusion would not be same. Bivalirudin illusion out of the column would not be same with that impurities. Means the compound is different, their characteristic would be different, and if so their illusion pattern would be different. So suppose if I am running the bivalirudin material in the chromatogram or in the column, they would elute at different retention time. Suppose one impurity present at 1% level in the bivalirudin would elute at 5 minutes. Within 5 minutes of time, this impurity come out of the column. But bivalirudin is not coming out. Bivalirudin is still struggling in the column and struggling to come out of the column. At what particular time bivalirudin have characteristics to come out of the column exactly at around 25 minutes, say for example. So 25 minutes exactly, if the peaks or the compound that is coming out of the column and going into the detector would detect that particular compound have come at 25 minutes, so that would be bivalirudin only. But there are other impurities which would elute at different retention time. So we can quantitatively estimate the amount that 25-minute illusion peak have, suppose say 95 percentage of the area. So we can say that the compound have 95 percentage of bivalirudin. Now what is present with other 5%? So we would say that 1% would be illusion peak at around 5 minutes, 1% at around 7 minutes, 1% at around 9 minutes, and 1% at around, suppose, after the main peak, that is say 30 or 35 minutes. So cumulative percentage would be 100%, and we can estimate that the bivalirudin content in the material would be 95%. I hope this is up to some extent is cleared in the minds of students. Now, this is the
Sir, excuse me,
basic.
sir.
Yeah, yeah, yeah.
We cannot see your current slide content.
Okay. Now is it visible, right?
Yes. Thank you, sir.
Okay, okay. So, coming to the analytical methods, now we have understood the classification of the instruments based on the purpose. What our purpose was to identify the compound qualitatively and to estimate the compound quantitatively. But when we understand the analytical methods, when you take any book or the theory, the classification of instruments is not based on the purpose, right? The classification of instrument is based on the principle. Now, what are the principles? So principles are, I mean, classical methods were gravimetry and volumetry, but latest and the conventional approach is latest current approach is instrumental methods. Now, instrumental methods, there are three principles again. First is based on electroanalytical parameters. Next one is based on electromagnetic radiation, and other one is miscellaneous methods, right? So these classification would be seen in almost any theoretical materials like in book or any other materials, right? So we also understand with this classification also why it has been defined in this way, right? We understood previously that qualitatively NMR IR mass is there, right? Quantitatively chromatography is there. So why is that? So it is based on the detection side, right? So if we talk about the instrumental methods, we'll first understand the methods, instruments working based on electroanalytical parameters. We'll also see some instruments based on electromagnetic radiation, and we'll also see some miscellaneous methods. Okay, so we'll try in this session, because this is very short time to cover all the things, but we'll briefly try to cover at least some instruments which work on electroanalytical parameters, and we'll also cover some instruments which work on electromagnetic radiation principle, right? So we'll start with electroanalytical parameter. Okay, so what is the principle behind these instruments is electroanalytical. If we know it, traditionally we were performing titration, right? So suppose if we talk about acid based titration, everybody knows, even students would also know that we are titrating base with acid and we work on the end point of the titrations. So these visual or practical procedure have been converted into electroanalytical. Why? Because whenever identify the electronic parameters in the compound, so that we can automatically identify the, I mean, instrument can automatically identify the endpoint and give the results to us. So the first automatic and the most used automatic instruments working based on the principle of electroanalytical parameters is the coulometer, Carl Fisher coulometer. There are three major instruments which are based on these electroanalytical parameters: coulometer, Carl Fisher titrator, and auto titrator. right? More or less all these three instruments have same structure, right? If you can see in the figure, if in the right hand side one blue color electrode you can see, right? Not the blue color wire, the attached with wire is not that, but the blue color rod glass electrode is there in the glass beaker, right? And behind that electrode is attached with a wire and going into the instrument. So the compound that we want to estimate needs to be inserted into this beaker and reaction can be started. Whenever endpoint would come, the instrument will automatically record the results and give it to us. Now, coming to the practical use of the instruments, in what way we are using the instruments which have principle based on electroanalytical parameters. There are two major purposes, and the first and most used purpose is estimation of water content. See, water content, we all know that water is an essential ingredient used in the chemical synthesis or formulation development. So water at least at the trace level or at the higher level, ranging from 0.1 percentage to 10 percentage or 20 percentage or 30 percentage in some hygroscopic compound, water is would be present in the material. And water also plays a great role in the overall potency of the compound, because water would increase, then the potency would decrease. So, and water also plays a role in degradation of the compound, because in presence of water compound degrades actually. Therefore, the scientist or the organic synthesis personnel always want to know the amount of water content present in the structure. So for that, this coulometer and Carl Fisher titrator instruments are used. The basic principle is same as we were doing titration, where we have to introduce the compound and we have to set the, I mean the reaction is itself is set, because there is vendor made already available in the market coulometer reagent is there. So that coulometer reagent would react with the water molecules present in the compound, and we would give it the reaction, and the result would automatically come that the compound, suppose 20 milligram compound I have inserted into the, I have dissolved in some diluent, and I transferred that solution into the coulometer. So that solution have 20 milligram compound. So the instrument result would say that your result, your compound have 10 percentage of water, say for example, right? Similarly, Carl Fisher titrator also work in the same way. Right now there is no scope to understand for you the difference between coulometer and Carl Fisher titrator. That would be out of scope for current discussion, but that is also a different, there is also a great difference between coulometer, Carl Fisher titrator, because of the amount of quantity used and all, right? But for current discussion, let us take that Carl Fisher titrator and coulometer can be used for water content determination of the material. Auto titrator, where we are use the first purpose of these instruments, we discussed that it is for water content determination. Next purpose is for assay, determination of assay of the compound. Right now it is not using much because we are estimating the assay through HPLC and other sophisticated techniques, but still there are some compounds which are difficult to get estimated by chromatography, and therefore still we this chemical reaction based estimation of assay is still there for some compounds which are not possible elsewhere. So for that, auto titrator is there. Auto titrator, same principle would be same like the traditional titrator, where we have to uh, we have to react we have to make the reactions between the compound and acid or base, and thereby we are getting some end point, and that end point would be the that end point would indicate that how much percentage of acid, how much ml of acid or base have been utilized to complete the reaction between base and the compound. And by based on the volume of the acid or base utilized, we can say that how much percentage of acid is present in the compound, right? So these are the three major instruments which can be which can be used for estimation, and they are based on analytical parameters. See, throughout our discussion, we will target two aspects. One is principle of the instruments, and second one is the purpose of instrument. This both way would definitely go hand in hand, right? So we have understand that these instruments are working based on the electroanalytical parameters. Now, and second purpose is estimation of water content and the estimation of assay of the compound, right? So I would try this within this 1 hour I would try to give you the way, right? Suppose if you want to understand more detail about the colorimeter, Kalfusser titrator or other things, then you can definitely have other materials and other things, so you can read and understand the things. But I am guiding you, I am showing you the way that the what are the instruments available, what is the based principle, and what is the purpose, so that you can move ahead with that after this discussion also. So moving ahead to the next portion of the instrument methods, and that is based on EMR, electromagnetic radiations. We now all of us have studied the electromagnetic radiations in pharma analysis in BPharm some course, right? So till date we were not knowing the exact importance of EMR, but I would like to tell you that this is the electromagnetic radiation, right? Starting from gamma ray, X-ray, UV, visible, infrared, microwaves, radio waves, long waves. Now, what is the importance of studying this EMR range? So, for every particular range of the rays, we have a set of instruments, and that instruments using that particular ray giving us the result, right? Therefore, these rays are important. These rays are important only because this rays detection. Suppose if we talk about UV visible, right? UV spectroscopy. So, if you can see in the figure, UV spectroscopy is between 100 and 100 nanometer to 400 nanometer, right? So there are compounds which can have chromophoric group, and therefore they can only be detected within this range. So what instrument would be used? UV spectroscopy, because UV spectroscopy can see that compound, those compounds, because those compounds have chromophoric group, they can excite themselves within this range, and thereby we can uh, we can detect those compound in UV spectroscopy. Suppose if I want to detect the compound which have radio waves, ultra high frequency radio waves in UV spectrometer, would it be possible? No. Why is that? Because those compounds have ultra high frequency radio waves. UV spectrophotometer is not capable to identify the compounds other than 100 nanometer to 400 nanometer. They would only UV spectrophotometer would only be possible to identify the compounds which have the wavelength region between 100 nanometer to 400 nanometer. So for every such range, we have different set of instruments. For X-ray, the compounds which have X-ray differentiation, we can use XRD, X-ray diffractometer. We have suppose compound have infrared rays differentiation, we can use IR spectrophotometer. Suppose compound have ultra high frequency radio waves, then we can use NMR spectrophotometer. So based on this electromagnetic radiation region, we have different set of instruments. So we'll study one by one other instruments based on this electromagnetic radiations, and we'll try to understand two aspects as I have said. First is principle of the instruments, and second one is the purpose of instruments. And the most important one, instrument we can find elsewhere, but the purpose I'm saying with you, I'm sharing with you, try to listen carefully, because that purpose would be difficult to identify it somewhere else, right? It would be definitely the extraction of my experience in the analytical laboratory, and that would be fruitful to you. So moving on to the next, first instruments, we'll try to cover one by one, starting from the right hand side to left hand side. We first discuss XRD, then UV visible, then IR spectroscopy, and then NMR, and lastly we'll discuss some HPLC and other aspects. Okay, so this is the today's discussion points. So we'll start with the XRD. So XRD is based on the constructive interference of monochromatic X-rays and crystalline samples. The interaction of incident rays with the same products difference when conditions satisfy Bragg's flow. Now, this is the principle of XRD. But what the purpose is, the basic purpose of using XRD is identification of the structural forms. Suppose if I have to take one example, I don't remember the exact drug, but some drugs have different forms. Say for example, ibrutinib is the drug, right? So ibrutinib drugs may have different forms, crystalline forms. There are some crystalline nature, amorphous nature, but out of crystalline also there are multiple forms: form A, form B, form C. So these are the these are the generation of response in the structure. How? Only technique which can identify the difference of these forms in the compound. Suppose ibrutinib have three forms. Suppose, say for example, well, crystalline form and amorphous form, right? So the compound is same. The only difference is physical appearance of the compound. One is in amorphous in nature, one is in crystalline nature. What would happen if I inject this compound into HPLC? The compound is same, ibrutinib only. So it would elute at the same retention time. It would not elute at different retention time. It would elute at the same retention time. What happen if I inject this compound into LCMS? It would give same molecular mass, not different. Form A would have the same molecular mass, form B would have same molecular mass. What happened if I inject or if I run this particular compound into NMR? NMR would also not differentiate between these forms, because both the forms have same chemical structure. So both the forms have same spectra in NMR. What happen if I run this compound into IR spectrophotometer? Again, IR would not differentiate the forms between A and B. It would give same IR spectra. So the meaning is, the only difference that we can see between different forms is using spectra. In XRD spectra, we have difference of the peaks, and that is the only way that can differentiate. So this is the purpose. And in industry, when you enter in, I mean, when you become a stakeholder of the industrial portion, when you get a job or you are involved in some pharma role, definitely you would have to understand the purpose, why this instrument is used, and what is the principle. If we understand the principle, then we will surely and easily understand the purpose. Moving on to the next instrument, this is the principle that we have theoretical way. moving on to the next instrument, and that is UV visible spectroscopy. Again, the principle that we all know, at least for UV visible, because it is available in academic institutions and we are using that in the colleges and all. So principle is Beer-Lambert's law. And the basic role of using purpose or the using of these instruments is the estimation qualitatively or quantitatively we are estimating this. Basically, nowadays the role of UV visible spectroscopy is very limited in the industry. But yes, UV detector is part have been made the part of the LC, because LC have UV detector. So UV photometer whole have been transferred into the HPLC, that we can say, because it is UV detector. So we have combined liquid chromatography with UV detector. So UV detector itself we are not using, but yes, chromatographically when the compound elutes into out of the column, we are making a provision that those eluents with the compound would be entered into the detector, and thereby we are using the UV visible spectroscopy in terms of the detector of the HPLC or other instruments. Next is IR spectroscopy. I already told that infrared light there are some IR active compound. See, for spectroscopy, this active compounds are told generally. When the compound is IR active, it shows different vibrational and rotational spectra, which is the characteristics of the functional group, right? So there is a, if I have the region, I would show you this. This is one. So this is the different stretch actually. Suppose ketone stretch would be only present in the region of 650 to 1800. So if I get the peak at between this region, definitely the compound have ketone group, right? So similarly, various functional groups have various peaks in the IR spectra. There is very this is one of the characteristics of the instruments, and based on IR spectra radiations the compound is giving peaks. I think NMR and IR, these are the only two techniques, and up to some extent MS, LCMS. So that these are the only three techniques where where we can where we can use where we can use these techniques to understand the presence of functional groups, right? So in IR spectroscopy we are clearly getting the peaks based on the presence of the chemical functional groups. In NMR also we have some idea about the presence of functional groups depending on the illusion of peaks, right? But in NMR we have to correlate the things. We are not getting clear peaks at the spectra like in IR. Suppose we are getting peaks at ketone position, ketone region, but in NMR it would not like that. We would have proton peaks, we would have carbon peaks. We have to correlate that the environment of proton and environment of carbon, and thereby we can estimate that this functional group would be present, right? But the exact idea, the the clear idea based on the IR spectroscopy is this. In MS also, the clear peak of the functional group would not present, but based on fragmentation pattern, suppose carbonyl group is there, so carbonyl functional group is C plus O, so 12 plus 16, so 28. So removal of 28, suppose the base mass of the compound is suppose 200, but the next fragmentation pattern of the compound would give the peak at around 200 minus 28, so that we can say that 28 molecular mass have been removed from the compound, so that we can estimate that carbonyl group must be present. So in this way interpretation would play a role. Let me tell you, when we are qualitatively identifying the compound, the interpretation of the spectra, maybe it is of IR spectra, NMR spectra, or LCMS spectra, all this, the interpretation of all of these spectra would definitely require a huge experience and skill, right? On one day, I mean, by reading the, or on one day if you learn the things, and on the next day you will start interpreting the things, that would not be possible. But yes, we can understand the principles, and by huge deep interpretation of the spectra we can understand the presence. So till now we have understood the IR, UV, and other spectroscopy like XRD. Now moving on to the next instrument that is based on electromagnetic radiation is Nmr spectroscopy, and it is one of the most important spectroscopy. And the instrument that you can see on the photograph is like this. If we talk about the principle and the instrument structure, we are using radio waves. We have generated the artificial magnetic field inside this huge white colored tank, if you are seeing, right? So in that tank, electromagnetic radiation or the magnet is there, and megahertz levels magnet is there, so which generates the magnetic field. And inside that magnetic field we are introducing sample, and we are, I mean, applying the electromagnetic radiation, so that the proton or carbon of the compounds can give different kinds of peaks. So this is again a detailed spectroscopy principles. This is based on the energy levels principles, and again, which kind of compounds are there which are uh, NMR active. So this is again a principle of the NMR, where odd number of the protons or odd number of the neutrons only would give the NMR peaks. Why is that? For that we have to understand the principles, because all that possess charge, and those with either an odd mass number or ordinarily possess magnetic spin, right? So oh, so this is again a principle, but let us, with time limit, let us move directly onto the spectra of how NMR spectra would look like, right? So here I have taken two spectra. One is proton spectra and other one is carbon spectra. So you can easily see that the proton spectra is of benzene ring, and the structure that I have drawn, right? And one position it is having phenol group, and at one position it is having CH2 CH3 group, right? So we got the proton spectra, where we are having peak at around 1 ppm level. If you can see the range of the X line of the proton spectra, 0 to 8 is there, right? So oh, so at around 1 ppm we are getting triplet peak, and that is due to CH3. Okay, CH3. See, proton spectra would give the peaks only due to protons present in the structure, right? So suppose if we talk about these structures which have benzene ring and the phenol group, right, and CH2 CH3 group, then the protons present in the structure would only be detected in the in the NMR spectroscopy, right? So how many protons are present? So CS3 group have 3 protons. Next to CS3 we have CH2 groups which have 2 protons. Then benzene ring have other 4 protons if we clearly and closely look into the structure. CH, 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 right? Two carbon of the benzene ring have been occupied by the functional group, so CH is not there. So four proton of the benzene ring plus CH2, two proton of CH2, three protons of CS3, and one proton of OH. So 3 plus 2, 5 plus 4, 9 plus 1 is 10. So total 10 proton are present in the structures. So it does not mean we'll get 10 peaks, but based on the amount and based on the structure we'll get some peaks. So CS3 peaks have been observed as triplet, and therefore at around CS3 we are getting some peaks. Based on CH2 we are getting quartet, and for OH we are getting some peaks. For CH, that 4 aromatic proton we are getting some peaks in the aromatic region. So how will identify the structure based on the NMR spectra? That is most important thing. So one thing is clear: the chemical shift, the the exact position of the peaks indicate the some characteristics in the structure. For example, suppose if I am getting 4 proton peaks in the region of 7 to 8 or 6.5 to 7.5, 6.5 ppm to 7.5 ppm is the region which is dedicated only for the aromatic protons. So if I am getting that protons in that particular region, I would definitely say that my compound that I have run would definitely have 4 aromatic carbon, 4 aromatic proton, right? So uh, I mean, do you have played that Sudoku, no? Where we have to think in different directions and we have to solve the puzzle. NMR spectroscopy, interpretation of NMR spectra is like that only, where we have to correlate different things. So first things I got that I have 4 aromatic protons. Then I am getting single peak at around 6 region. So single peak is the characteristic of OH, NH2, NH likewise. So I would think that my compound must have this kind of two to one out of this 2 to 3 groups must be present. There is the region dedicated for aliphatic chain, and that region is 1 to 3, between 1 to 3 ppm. So if I am getting the peaks in NMR proton spectra between the region of 1 to 3 ppm, I would definitely say that my compound have that particular protons in terms of aliphatic chain. So again the splitting pattern would come. See, these are the aspects that would definitely be covered in the detailed interpretation segments of the NMR spectroscopy. I run one session based on these NMR spectroscopy interpretation, and where there I discussed in detail about these aspects. Why, what is splitting pattern, why this splitting pattern comes, why this CH2 proton peaks have occurred in quartet shape, why this CH3 have occurred in triplet shape, what we can estimate, right? So these are the things. But as of now we can understand that in this way we can extrapolate our knowledge of NMR spectroscopy to identify the structure or to confirm the structure, right? Similarly, proton spectra would give the peaks only due to proton. Similarly, the second figure in the figure is carbon, carbon spectra, where we are getting the peaks due to carbon of the compound. See, most organic compound, the most abundant amount of the molecules present or the atoms present are carbon and hydrogen. So we are estimating hydrogen in the proton spectra, we are estimating or confirming the carbons present in the structure using carbon spectra. So remaining would be assumed, and remaining would be can be correlated, right? There is also detailed cozy and nosy analysis where we can combine the carbon and proton spectra one to another, x and y axis, and we can correlate the presence of carbon with the proton, and likewise this is again a very detailed study, right? So as of now we'll understand this. Now moving on to the next some aspects related to HPLC and UPLC and other things. So this is some of the photographs that you can definitely see these instruments in almost every industry, right? This is the basic level of instruments, HPLC. Okay. Without this, no laboratory, not a single or not a smaller level of laboratory would run, right? In these instruments, if you have, you can see there are mobile phase bottles in, and there are column compartments on right side, sample holder, you can see the door. On the upper side there is a portion called detector, UV visible detector is there. So basically mobile phase is coming down and entering into the column and coming out and going into the detector. So this is the flow of instruments, right? Again, HPLC have been transformed into UPLC. You can also see these instruments. It is ultra high pressure. Now, let me tell you one thing. HPLC, this is high pressure liquid chromatography, which bears the pressure of 5000 psi. When we are running eluents into the column, no, it creates a pressure. So that pressure is up to 5000 ppm. So for 5000 ppm we have a limited utilization. We have to keep the longer run time, and we have also have to inject the higher injection volumes. So next technology is UPLC, ultra high pressure liquid chromatography, which bears the pressure of 15000 psi, so that we can inject up to 1 microliter 2 microliter injection volume. We can also have smaller columns, and we can also have shorter runtime, 10 to 15 minutes, and sharper peaks and better chromatography. So this is again the next level of instruments. This is typical LC chromatogram that all you. Sorry, my battery is running down. Let me connect to the charger. 1 second. Okay. So this is typical LC chromatogram in which LC, there are various peaks. Uh, this is again a detailed analysis. We can see the different compound have different illusion. That is the basic principle that we should keep in our mind, right? See, these are the chromatogram of parabens, and various parabens, though they are structurally related to each other, they have different chromatogram, right? So this is again the photograph of LCMS. See, LCUV, we have a UV detector. But when we talk about LCMS, HRMS, we have replaced UV detector with MS detector. Why? Because the principle of MS, we want to identify the compound which are not detected by the UV detector. So we can ionize those compounds, the compounds which don't have the chromophoric group. We can easily ionize those compounds and detect into the MS detector. So it is 10:55, and I think we are running short of time, but we have a lot to discuss. But if I cover in a little bit faster manner, we'll quickly discuss this LCMS spectra and we'll finish it up. So in LCMS spectra, suppose this lower picture suggests the peak, and in LCMS that peak, the eluents that come out of the UV detector would be entered into the MS detector. In this figure, if you see the right hand side, big structure is there, and that is MS detector. So HPLC bottle mobile phase comes out of the column entering into the UV detector and then entering into the MS detector, so that it can be ionized and detected the exact molecular mass of the compound. So in this way we'll get, suppose in the first spectra we are getting 291.2 and 281.2. So these are the two characteristic molecular mass, right? So in this way we'll have some masses. This is again a detailed understanding of the LCMS spectra, where how can we, if we have a future in suppose detailed analysis of LCMS spectra interpretation, then we'll have this discussion how singly charged molecular mass would come, how doubly charged would come, and what is the difference between that. And that's all from my side. That is a lot more to discuss, but since a lot of time is there, I would better have some query, then we can have discussion on that query. So if any one of you have some questions or concerns, you can definitely discuss with me. We'll have discussion. I would happy to sort out all the query.
Thank you very much, sir. Actually, we are running short of time. So, whatever questions students may have, we will ask you through email. I have your
Ah,
email ID.
sure,
So, we'll
sure.
surely clear all the doubts. Right, Asa madam? It's already 10:57. So, thank you very much, sir, for enlightening the audience with very informative speech for instruments which are used in industry right now, the real application of the instruments. And on behalf of Parallel Institute of Pharmacy, Parallel University, I thank you webinar team also for their support. I would like to thank management, Parallel University, for providing us platform for conducting the webinar events. And I heartily thank Dean, Parallel Institute of Pharmacy, Dr. Abhay Dharamsi, for encouraging us, for arranging this type of expert talks. And lastly but not least, I would like to thank all the teachers and students who have actively participated in this webinar. Thank you, everyone. Have a nice day.
Okay, thank you so much.
Thank you, sir.
Okay.
Asa madam, with this, we'll conclude the webinar here.
Yes, madam, it is over.
Yes, sir, it's over
Okay,
just now.
okay,
Yeah,
okay.
thank you.
You can exit.
Yes.